Okay. Alright, so we get started with the PTM tutorial. We'll go through some of the things that we can do specifically with relation to PTM data. This is obviously geared towards phospho because that's the data set we're working with, but many of the steps are generic, so should be reasonably applied to other PTMs. So, of course, you'll need Perseus. I would recommend, if you haven't done this already, to copy the data set onto your local drive so that you're not accessing it from the USB stick. So the first thing you'll need is in the assignments folder, this modifications step-by-step sort of in guide. And then in the data folder, you'll find practical 3, the phospho STY sites table. So in Perseus, you should click this green button, the generic matrix upload, and go ahead and select that file. And what you'll see is, as you've already seen in the previous tutorials, are all the columns on the left that you can select to be loaded into your matrix. And for this tutorial, we want the intensity columns because it's label-free phospho data. Here we have 6 control samples and then 4 for each of the 2 conditions, which, as you've heard from Kirti, is EGF and nicotazole. So in this case, because it's phospho, we have the phospho STY table, we have _1, _2, _3 for each experiment that was defined when max quant was run. And these, as you've heard, correspond to the singly, the doubly, or the triply, and higher phosphorylated peptides. So these are the columns that you should select to load into Perseus. Before I do, and you can see that they're actually, to do that is, in this example, a little bit laborious because we have to, go through and select just these ones. But I'll just show you, if you use the latest max quant, along with obviously loads of other great changes, one of the things Jürgen has implemented is grouping. Okay, it's not necessary, but if your data is processed with the newest max quant, what you'll see is that rather than being split like they are in this case, you can just select all of the intensity _1, 2, and 3 and drag them across in one go, because they don't contain these mod over base in between them. So they're grouped such that you can select them much more easily now. So anyway, go ahead and you can select these intensities here. Okay, so what we also want to make sure we've got, and it's already been automatically selected, is the localization probability column. So that will become useful. So I'm going to load those up. Sure. Okay, so now we can see the columns loaded into the expression matrix. So as with most data sets, probably the first thing you want to do is remove the reverse and possibly the contaminants, whether or not it's relevant to your situation. In this case, I think it's highly unlikely that keratins are going to be important in this study, so I'm going to filter those out also. So we go to filter rows based on categorical column, and there you can select reverse or contaminant, and you want to remove matching rows. And something you'll notice over here, we have this status, all this information about the matrix, and if we switch between matrix 3 and matrix 4, you can see the number of rows here has been reduced. So that's a useful tool that you can use to see whether what your filtering has done to the data. But we can also see, if we go now to the reverse column and click on it, it sorts, and we can see that there are no entries there. So it's worked. So let's do that again. Slow down, sorry. Okay, yep, no problems. Yeah. All right, sorry, it's filter rows and filter rows based on all right, great. So the next step is just to filter out. Right, the localization probabilities should go into the numerical columns, but it should be selected by default usually. Right. Yeah. Okay, so generic matrix upload.
What was the, after these intensities?
Yes.
What was the
Last one
the other thing?
he's redoing it. Yeah.
I changed the resolution on the, yeah. I didn't do, I mean, anything else, so it must be it. Okay, is everyone? Yes, so it's here. If you see where the mouse cursor is, under numerical, it's selected by default. So I'm just saying in case it's not, to make sure it's... Okay, I'm not going to do it again because I've already done it in the previous matrix. So we now have the data loaded. Okay, so is everyone? Yeah. Okay, so filter, we want to filter the reverse entries. So filter rows, and filter rows based on categorical column. No, filter rows based on categorical column. There wasn't I mean, I didn't filter that yet. Okay, I should point out that with Perseus, although it's a linear workflow, we don't necessarily have to follow the steps in any particular order. So we can filter, as in, I'm sorry, in the thing here, I think we're filtering out the class 1 sites first, and then I just now went and did the reverse first, but it doesn't actually matter which one you do first, and that's an important thing to take home from this, that certain steps need to be done in the right order, but most don't. I mean, whether we filter the contaminants out first, or the reverse out first, or the poorly localized sites out first is not going to affect the matrix. In the end, it's just a filtered matrix. So you can, so in that case, maybe follow along with me rather than the manual here specifically, because some steps that I'll do will be faster in the order shown live, rather than, and if you want to do it later on, you can follow along here. But there's the same steps are in here that I will do, not necessarily exactly the same order. Okay, so we've filtered the reverse. Now, right, filter rows based on categorical column. Then select the reverse in the column that you want to filter. And then you want to remove matching rows. Yeah? So these are the same steps we did yesterday for the proteome data. So now we have the matrix without the reverse entries. We're going to do the same now for the contaminants. So filter rows based on categorical columns. Select contaminants. And remove the matching rows. Okay, so now we have those two columns, reverse and contaminant, but still in there, but they're empty. So we can go ahead and remove those if we like. We can go rearrange, and then reorder remove columns. And then you'll see down here categorical columns where there's reverse and contaminant. If we move them across to the left, then we will no longer have them in the new matrix. So go ahead and do that. Okay. So now if we have a look at what columns we've got, there's no reverse and contaminant columns anymore. And in the same way, you can reorder columns. Well, yeah, I mean, it's just sometimes not nice to have empty columns, but it's really just to show you guys how to do it, because it hasn't been covered. So now the same as yesterday, also for cosmetics reasons, we may want to rename the columns here. I mean, they're already a bit, they're not so nice looking. So let's go rearrange, rename columns with regex. And if we type in here the string which is repeating, in this case it's German. Intensity dot star, intensity space, and then in the brackets dot star. And if you hover over regular expression here, you'll see a little tooltip which tells you, actually they say in this case ratio, heavy over light, normalized, dot star. So they give you that example to remember. Okay. So if we do that, you'll see now we've removed that from all of that part of the column header from all of the columns, so they're easier to read. Okay, so now we'll filter the data for only the class 1 localization probability sites. So for that, we go filter rows. And now we want to filter, instead of on a categorical column like we've always done before, we want to filter on the numerical or expression columns. So if you select that, you'll see this the filtering options. And so the number of columns, we just want to filter one column. The column that we want to filter is the localization probability column. And the rule that we want to use to filter it is x is greater than 0.75. We want to reduce the matrix using that rule. Okay, so now if we switch between matrix 8 and 9, or the last two matrices that you've got, we'll see that the rows was 59,000 and now we're down to 41,000. So you can, using this tool, this information here, you can see what's been applied to your matrix. And in case it wasn't clear, as in previous sessions as well, you can also click on the functions which you applied to the matrices on these, and you can see exactly what the settings were that we used. So in this case, we filtered rows based on a numerical column. It's up here, and we can see all the settings that we put in. So in case you forgot what you did or you weren't sure, you can go back and check, did I do the right thing? Okay, now the next thing we usually want to do is logarithmize the data. So you do that with this transform button, but it's also under basic. So if you go to basic, transform, and press OK, you get the log2 of the data. And this will, in the case of zeros, fill the column with NaNs, which will become handy in a minute. So the next step in the instruction guide is to expand site table. I'm going to leave that for the moment because this will, as you'll see, it will increase the size of the matrix considerably. And I don't want to do that just yet, so we don't slow down everyone's computers. We've transformed it, and we've filtered already, and we've done the regex. So we're basically now up to page 4. So to add the annotation, we use annotation columns, add annotations. So this will add the protein annotations for each of the rows. So it's as you'll see in this function here, it's using the, okay, so here we don't have the, as you may not also have, the, we haven't got the correct source, annotation source. So that is located under annotation on the USB stick, in case you haven't got it. It's human, this data set, so we want this one here. So copy that and put it into your software folder. Perseus, config, annotations. So place it in there. And while we're there, we also want off the USB stick, again under annotations, inside this PSP folder, so this stands for Phosphocyte Plus, we want the kinase substrate dataset and the phosphorylation site dataset. So if you copy those and put them into your into your Perseus conf folder, you'll find the PSP folder. So put them in there. exactly, kinase substrate dataset and phosphorylation site dataset, because they're the ones relevant in this practical. Okay, so I'll just do that one more time in case you missed it. On the USB stick, there's a folder called Annotation. In there, you'll find the UniProt annotations, which are protein annotations, and we also have this PSP folder. Inside the PSP folder, there are some data sets from Phosphosite Plus. So you want to put these ones into your PSP folder and the UniProt ones into your annotations folder of Perseus. It doesn't matter, I mean you can take them all, but the ones we'll be using today are kinase substrate and phosphorylation site dataset, but you can put them all there. We good?
Hello.
Right.
Yeah, my question is, why do you have a single probability even though there are 3 different sites?
Right, so when you load, that's a good question. When you go generic matrix upload of a phosphor STY table, you'll see actually that we do have a localization probability for each one of them. But in this case, we just use the global localization probability.
So is it like of the 3, whichever is the highest one is being shown there?
Was it the highest one is being shown? Is that the question?
Yeah, I mean, if you have 2 sites and one is 0.75 and another is 0.3, so it's only showing the one which is the highest one?
It's actually the lowest.
So actually this is a site-centric table, and what you're seeing is you already filtered your class 1 sites here, right? And this has to be distinguished from, I actually don't know who asked the question, but yeah. So it's a site-centric table, and you may have it quantified as singly phosphorylated, doubly or triply, but that information that you're talking about is sitting in the modified peptide table. You have to understand this. This is the site-centric table, which is giving you quantification on these different instances.
Okay.
Right.
Yeah.
Yeah. But in answer to your question, it's the highest in the experiments, right?
Yeah, in this case, it's the highest.
The global localization probability reported is the highest, and then quantitative information will only be shown in the experiment if the localization probability is greater than 0.5 for each experiment. Okay, so now we have log transformed our data. The next step we can do is to create groups, and the reason for doing this at this point is that we can use the groups to perform filtering. So no, we haven't added annotations yet. I mean, right, okay, sorry, that's what we're up to. Right, so annotation columns, add annotation. So as I said, this is the protein annotations. And here now you should see the Homo sapiens annotation. And just go ahead and add down to pfam. I think in the instructions I've said down to quorum, but you know, we don't trying to keep this running on your laptops. So let's add fewer rather than so you take those across. Okay, that probably or may not be finished on your laptops, but I'm going to do the next step and then I'll repeat it because I want to get the next step started on my laptop on here. So the next step is to add modifications. There are several things in here that we can add. This is now modification annotations. So previously the step that we've just performed and we're all waiting for was protein annotations. Now we have annotations that are sequence specific. So for example, we can add linear motifs. And these sequence specific annotations relate to, they use the sequence window, which is over here, which you'll find here. So this is the sequence window for that site, and what you'll see is that the central residue is the one that was modified. The surrounding residues are coming from the UniProt sequence database. In the case that you have an N-terminal or C-terminal modification, just see if I can find one. In this case, we were close to the N-terminus of the protein, so you see that we still keep the same length of the sequence window, and we instead put underscores there, just so you know. That's what that means. So modifications, add linear motifs, and the sequence window is already selected. That's the one we want. Sure, yep. So after the protein annotation, we've just added the linear motifs. So that was modifications, linear motifs. Are we mostly good? Okay. So we can also add now sequence features. Right, sorry. So if we go to the protein annotation, I'll show you what was added first. Here's the Go terms and the KEG terms and the PFAM terms. Now these are, because as I've said, they're protein specific. They'll be the same terms for each. If 10 sites come from the same protein, they will have all the same annotation there. The second thing that I added was the linear motifs. And that adds this column here, motifs. In case you wanted to define your own motifs here, the way that it generates this annotation, this sequence-specific annotation, can be modified by you. So you can go into, if you go to the USB stick, into Perseus, into the config folder, you'll find this file called motifs.txt. This is the way that this annotation is generated. So if you open this with Excel or something similar, I mean, it's not, apparently we don't have Excel. Yeah, we do. Yep. Yep.
Sorry if I missed this. So when you pull in the data from Phosphocyte Plus,
Yep.
is it organism specific?
No, those
Can you-
data sets aren't organism specific. I mean, no.
So if you were, so you're just pulling in all data, human, mouse, rat, that's on there.
No. Okay, so that motifs.txt, if we open it with a spreadsheet program, you'll see what it consists of is the annotation down in the first column, and then in the next column it has the sequence preceding the modified residue. Okay, and single letter amino acid codes, and X means any amino acid, and they're separated by commas. And in the case where you want to offer several options for an amino acid, as in here, you just put them all in a row without a comma. So just to work through an example, this here means that the first residue in the sequence window would be an R, followed by any residue, followed by another R, followed by another 2 residues. Then this column here is the central residue, so the modified residue. So in this case it's either an S or a T, and then the last column here is the sequence that's after the modified residue. So in this case F or an L. Is that more or less clear? Yep. Microphone. Yes? Yeah.
Is this from the Human Protein Reference Database?
Yes.
Okay.
Yeah. Okay, so I don't want to save changes to that. Okay, so that's that. Other annotations we might add are, for example, sequence features. So I'll show you what they are in a second. For this we need to define our proteins column, so that's the UniProt identifier, and the position of the modification within protein. So the columns are usually selected correctly, but in case they're not, then you can fix it. Okay. So then we see all of these sequence-specific columns, such as whether the site occurs in a zinc finger region, signal peptide, etc. So just to get back on the same page, we're now more or less finished page 5, and we're on to page 6. So on page 6 we want to define experimental groups. And that's done using annotation rows. There's also a button just here, Categorical Annotation rows. So if we go and do that, just as with the total proteome, we see that we can create groups for our experiments. So in this case, we didn't have too many experimental groups, but that already became quite tedious. So just as yesterday, you can you can use a template file, or you can create from experimental name using regex in case you want to do it, you know, in a more efficient way. So if we go ahead and create those groups, what you'll now see in the matrix, I'm not going to continue, I'm just you'll see the groups along the top here now. So I'll just show you the other, some other ways we could do that. Nope. It's not going to work anymore. Okay, so we can use these groups for filtering. Under filter rows, you'll see filter rows based on valid values. So a lot of the columns in this matrix now might be empty or have very few values. But also remember that we loaded them as _1, _2, _3. So before we do filtering, we're going to expand the site table now, as Kirti explained, so that each row is a singly, doubly, or triply phosphopeptide. So to do that, from modifications, we go to expand site table. Wow. I've still got this computer here. I don't know. Is there a reason? It's weird. It's weird. Yeah, I think it's him. Okay, for some reason they've switched it to the PC here or something, because I've still got this one. Okay, thank you. Alright, so we just expanded the site table. We did too. And that took a while. Okay, well, yeah, maybe some microphone. Jürgen? Jürgen, use the group experimental groups when you expand the site table? You lose
On the
them.
site table expansion, I got an index out of range
That be-
error. Do you
I mean,
know
they
how to
should
fix
retain,
that?
right? Sorry, yeah.
On the site table expansion function,
Yeah.
I got an error message that says index out of range. I've gotten this for a couple of other functions in the past. I don't know how to
Okay.
see.
I mean, that might be a computer specific thing. Okay, so we lost the I wasn't aware of that. We lost the grouping there because we expanded site tables after creating the grouping. So
Okay.
apologies for that. So we need to then define the groups again, but at least now we have 1/3 the number of columns. So annotation rows, yeah, you can see this is not, I mean, I'm trying to do this in the way that you would work. You don't sit there and follow some sort of a flow chart. You just, this is how you can work in Perseus. You think logically, what do I want to achieve, and then how to get from A to Z. So now we have a much more reasonable number of columns here. The important thing is here that is each one is named the same thing. So it could just be C, 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 E, 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 and N, 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 if that's faster for you. Okay, so hopefully now you have experimental groups again for your site, for your expanded site table. Something I should point out to you is, you know, the effect of that site table expansion. If I go to matrix 15 on my screen, which is the one before I expanded the site table, we can see the number of rows was 41,000. But immediately after expanding the site table, it's 120,000. So basically we tripled the size of our data there because now we're working with single, double, and potentially even triple phosphopeptides there. But as a result, we will have many rows which, I mean, we don't have single, triply phosphopeptides for a lot of the sites. So we'll have a lot of rows which contain no values at all. So now that we've created, I'll just show you where they are. There's also, after expanding the site table, a new categorical column called multiplicity. So the multiplicity column is now just what was within the column header before, _1, _2, _3. So if we scroll down, we see all the single, double, triples. Okay, so we definitely need to do some filtering here because we have a lot of empty values. So if we go to filter rows based on valid values, we can see now that we could just filter in the total matrix, but we can also use in each group or in at least one group. So in at least one group is particularly useful for label-free and especially even for phospho data, because if you can imagine, if we have treated one of our experiments with some stimulus which has increased the phosphorylation dramatically, then we might find a lot of sites quantified in that condition. And even with matching between runs turned on, we may not be able to identify anything in the control samples or in the other samples, simply because it's many orders of magnitude lower. So in that case, if we just filtered, if we required to have 4 valid values in each group, like I've got selected here, or 4 in each group, then we would potentially throw out a lot of our regulated sites, because now we're requiring them to be in both the control and the stimulated cells. So instead it might make more, I mean, you need to think about what makes most sense for your data and try different things, but it probably might be worth trying to filter in at least one group, but rather require a large number of values in at least one group. So in this case, in the EGF and nicotazole treatments, we have 4 replicates. So we're going to require that all 4 of them have valid values. So we're really confident in the quantification in that sample. So the grouping is the group that we created just now. We want them to be valid, in other words, not NaN. And we want to reduce the matrix. We want to remove the ones that don't meet these criteria. Okay. So you see now, you will see in the new matrix, the number of rows is now 40,000 again. So we've gone from expanding the site table out to 120,000, and then we filtered and we have a much more reasonable number of valid values to work with. You can see here the table is nicely populated, the matrix. So at this point, it probably would make sense now to have a look at how our data looks. So we want to do some QC. So for that, we often use a histogram. So that can be found in the visualization menu, histogram. So we've logged, transformed the data already previously, so it should be, we're hoping that it's nicely normally distributed. Let's have a look. Okay, so when you do the histogram, you get a new tab along the top here. And indeed, when you do any visualization tool in Perseus, the matrix that you applied it to now gets these supplementary tabs, the visualization tab. So if you click along the top here, you'll see the histograms that we just plotted. So this is particularly useful to determine whether or not we should apply some normalization, as Kitty was saying. So, you know, maybe one of our samples was less intense than the others and needs to be normalized. In this case, you can see the data is well centered. It looks pretty good, so probably you wouldn't need to do any normalization. But I'll show you how you would do. A simple tool you might want to do in case these weren't looking centered. So you would go to normalization and subtract. Because the data has been logged, we want to subtract the median of the columns. And then press OK. So then the median of each column has now been subtracted. And we can again plot a histogram and see what effect that had on the data. So this was the histogram before. This was the histogram after. So you can see the data is now centered around 0. Okay, so having normalized our data if we determine that it's necessary, and checking what results, what effect that had on the distribution of the data, that we didn't cause some sort of strange artifacts, you now could consider imputing missing values. We've already filtered out. The important point here is that we're not imputing a large amount of data, because we already applied some very stringent filters to the data. So we said that we have to have 4 valid values in at least one group. So you definitely wouldn't go and impute without filtering first. But to do the imputation, you click Imputation, replace missing values from normal distribution. And the default values are generally working very well, but you can play around with them if you want. And of course you can then plot a histogram and see what effect the imputation had. So I'm going to replace the missing values. Yep.
Those values are?
Right, so it basically, as far as I know, Jürgen might correct me, it takes the distribution of each column, and then it and then it downshifts by this parameter, and this is the width of the distribution. Right, so I'll show you with the histogram. But basically, I don't think there's a logical way to choose those values. You just try, larger ones and smaller ones and see what works best. But if I plot a histogram, I'll be able to show you what the downshift means. So hopefully you remember from yesterday that we have these underlying matrices. Quality was one of them, and another one was is imputed. So now, because we've done imputation, we will have a matrix underneath called is imputed, and some of those will be populated. So we can't see that, but when we go to a histogram, if you see up here, you've got selection from table, and that would mean if you selected individual proteins, or sites in this case, from the table, but we want to go to select from imputation, and there we can see the imputed values, and you can see that they're downshifted and they have a narrower but the same shape as each column that we imputed for. So the assumption here is that the sites which we weren't able to match between runs were most likely low abundance in the sample. So therefore it makes sense to impute a distribution that's lower than or on the tail of the population.
I have a question.
Yeah.
Is this kind of imputation a standard in any field of bioinformatics?
I mean,
No.
well, it's probably a less well-known one, so it's. I mean, it's not a standard in any sense, but it's something.
Jürgen, talk it louder.
Hello?
It's really quiet.
So it's not a standard in any sense, but also, what would you mean with a standard? I mean,
Nobody heard you.
okay. Do you hear me now?
Yeah.
I mean, it's not a standard in any sense, but I mean, what would be a standard? I mean, what?
Yeah.
You mean frequently used, or what?
I don't know. Yeah. I'm just wondering, because imputation is a risky business, right? And
So if you want to use a standard thing, then I guess what is most frequently used is k-nearest neighbor imputation, but that works really, really bad. So I mean, we just came up with something that works really very, very well for our kind of data, and we highly recommend using that.
Yep.
Is this described in the literature anywhere, this kind of...
So that's something that we came up with, and I think we don't have it. Do we have it in any publication? I don't think yet.
Yeah, we should.
Used, but not used, but not described. I mean, the description is very simple,
description
right?
It's described, but it's
Okay,
it's
if I go back
described
home and start this imputation, I'm going to get into trouble with a lot of people.
no, but I
no
mean the
ability
important thing here
to
is, as I
defend
said,
myself.
you're not. The important thing is that you're not imputing large amounts of data, and it's also understanding where how the data is generated. So in this case, we know that the match between runs was used, right, and it should transfer identifications provided that the MS1 feature was present. So if the MS1 feature is not found, we can reasonably assume that it was low abundance for some reason, or for some reason it was missing in the sample. So therefore, it makes sense that if we were to impute that point, we should choose a downshifted value.
Of course, yeah,
right?
That makes sense. But
so, I mean, I can't give any, I don't know, we can't give any guidelines for what you should do with your data, but the important thing is that you would filter stringently first, I think.
Yeah, but so maybe in terms of possibilities of getting into trouble, so you won't get into trouble because the algorithm would be, because it's really extremely simple, the prescription, right? It's just taking the mean and standard deviation, and then you make a new distribution with these kind of prescriptions. So it's extremely simple. So I mean, nobody can say it's not. I mean, one cannot understand what's going on there, right?
yeah, so I can describe.
Yeah.
Yeah.
Yeah, I mean, it's also much, I mean, if you think about what other options, Jürgen said we could k-nearest neighbor, but you could also just fill it with a low value. And that option is there in the imputation, replace missing values by constant, and you could choose a low value, but that's much more likely to give you spurious results in your, statistical tests. So this is what we've, this is...
Yeah, so the problem, so one could use k-nearest neighbor. The problem with this is that this really works very badly for things that are completely present in one group and completely present in another group, right? So the k-nearest neighbor would look for correlations within the group where it's present, but that what it finds then, of course, not markers for the distinction between the groups, right? And that's a big problem with k-nearest neighbors. And so, I mean, if somebody is critical about what we are doing here, one can just compare. I mean, one can just also do k-nearest neighbor and will find that it works much, much worse.
If we were to do this again and again on the same matrix, would we get slightly different values? Is there a random seed then? Okay.
Yes.
Yep.
So if I can just pitch in, because we've discussed this a lot internally, and I frankly don't think there is a really a right way to do it always. What we were thinking is that if you think that your missing values are completely at random, then it might be better if you just take random from the normal distribution without any adjustment factor. But if you have something like, if you're hitting your cells with a sledgehammer, LPS or something, then probably you're missing values because of the abundance, and then you may want to do just, pick from the war tail, basically.
Okay, then then if you wanted to do that, you can adjust the downshift. So if you don't want to downshift, just set that value higher.
So maybe I can also give a short comment. So usually the imputed values serve as a reference base. So you need something that you want to compare to. You want to judge whether your site is above noise limit, or if your protein's enriched or something like this. So it always is something that you compare to. So in the end, you just need to make sure that you don't call anything significant based only on imputed values. So the imputed values themselves should not end up being significant, but something compared to the imputed values might end up being significant.
Yeah, and that's what we're using it for here.
I just want to add another comment on this. So basically, this is not a magic button to click. if you have a time course data, then I would say fit a curve and impute based on the curve fitting will be much better than the k-mean, k-nearest neighbor imputation. So basically, it really comes down to what kind of information you have in your data. If you have more information, you can use those information to help you to impute.
In principle, yes, but there's a big danger in that if you use information from your data for imputation, because then if you go on and use other statistical tools on that imputed data where you used a lot of information already, you're bound to overestimate p-values, significances, and so on. So best example is if you use the grouping, so you have like a two-group situation, and now you use actually the grouping information when you impute, right? You can do that. Of course, also the data looks much better than if you do that. But if you now go on and calculate t-test p-values on this, then they will be completely wrong, right? So that is very dangerous if you use information from the data, from the experimental design in the imputation.
Is there?
No one's busted.
I had a question. So this was intensity data.
Yep.
Would you do the same approach with SILAC ratios?
No, and that's actually one of the nice things about using label-free for phospho, is that, I mean, it's you can't impute ratios. So that's one of the things that we like about this workflow, is that missing values become less of a, you know, complete halt situation. Can deal with them at least to some degree more easily.
So just to comment on that, so if you're using SILAC as an internal standard and you invert the data, meaning that actually you're looking at ratios L to H and basing on the fact that we do match between runs, then actually you can assume that where you didn't find a ratio value, it is because the light intensity, so basically the experimental sample is very low, because otherwise, so it means that both the heavy and the light are low, meaning that you can actually assume that it's low, and then we are imputing. So the values might be different. So I think usually the downshift that I use with SILAC standard is lower than the value that I use for label-free, but still we do it, and the assumption should still be correct. Of course, if you're using classical SILAC, it's irrelevant. Let's put it this way.
Okay.
I have a question. Could there be a function added that you can see actually the values which are significant just from the imputed values? I mean, especially if you just select now, you demand to have 4 valid values in just one group. You have then 8 imputed values, and it happened to me that then I had significant values just from the imputed groups. So could there be easily a function added that you said they are kind of taken out of the significant values or highlighted?
Yeah, well, that's the nice thing. Now we have this underlying matrix with is imputed, so that could easily be. I think that's broken, that one. Just need to shout. That's.
Yeah, so it's possible already now, because so you can, after you have done your testing, you can set the imputed values to NaN again, back to NaN,
right.
and then you will see that.
Oh, that's good, yeah.
So we have here in imputation, replace imputed values by NaN. So now if you go and do that.
Sorry, I had a simple question. So when you're choosing your downshift, what exactly are you looking for to make sure that it's the right? Because you said you just kind of make it bigger and smaller. What are you trying to, how do you know it's right?
Right. So you're basically looking for not creating an entirely second population, I think. I mean, yeah, I've highlighted, I've shown what she's talking about, so we can see what I think the question was what you're looking for with the downshift parameter.
Yeah,
I think
how do you know
Marco
you
is
need
best to answer this because he's.
Yeah. So in this case, the fraction of imputed values is rather high. That's why it looks it's bimodal, but it's actually not an entirely separate distribution. So in case because you see that there's a little bit of blue on top of the red hill, right? So in case you choose the downshift too large, then the red population would be entirely separate from the blue population. In this case, you could just have imputed very low values because you will always get significance in case you compare something that was measured with something that was imputed, in case it's completely separate. So they should not be separate, but of course the imputed value should sit on the lower end of the distribution of the measured values. And usually the predefined parameters of 1.8 standard deviation downshift should do a pretty good job, and then you can play around a bit in the range of, let's say, between 2.5 standard deviations to 1.5 standard deviation downshift.
Okay, if you preferred to be more conservative, I'll show you what a downshift would look like on, you know, even more stringently filtered data, but don't go and do this because it's not necessary. I'm just showing you. So if we filter based on valid values now, and we require in each group 4, so now if you remember, the treatments have 4 replicates, but the control has 6. So in this case, the only missing values, there's none in each of the treatments, but in the control there can be 2 missing values out of the 6. So I've just imputed and plot a histogram. And as Marco was saying, you can, you know, we want this imputed population to be ideally part of the same distribution, only downshifted. And that's what you can see here. Only there's much fewer imputed values. So you can see that might be useful to convince you that we haven't shifted it entirely away from the existing distribution. But as I said, if you did that filtering, you could now lose incredibly significant populations of sites. So I mean, it's up to you what you do, and I'd say try different things and see what works for the data set. So I'm going to move on. Just using the workflow here, in case you haven't seen it, you can select many nodes at once, and you can delete them. Remove selected nodes using that button, and you can delete them. So this, you know, can be useful in case your workflow is getting really busy. So moving on, we filtered, plotted histograms. We can check the correlation between the data now using Scatterplot or multi-scatterplot. I really wouldn't recommend you run the multi-scatterplot because your computers won't get through it in a reasonable time. But so we can just do a single scatterplot. Okay. And here's where you, just the same as yesterday, where you can check your correlation. Okay, so now we want to perform some sort of significance testing on the data. So for that we go tests, ANOVA, multiple samples tests. And in this case we're going to use 0.001 because the treatment that was done was caused a very significant change in the phosphorylation status of the cell. So mitosis, you know, we have, it's widely known that more than half of the phosphoproteome is most likely drastically changed by this treatment. So we're going to we're going to put an FDR of 0.001 and an S0 of 1. Okay, so you will get now an over significant column. And as I said, this treatment caused really drastic changes to the phosphoproteome. So we have a lot of significant columns, and we want to now see what's happening in terms of expression profile or phosphorylation changes of those significant ones. So we want to filter and keep only the ANOVA significant rows. So for that we can again use the categorical column filter, just as if they were, you know, reverse or contaminant. But instead of with reverse and contaminant we wanted to throw them out, this time we want to keep them. So if we go down to ANOVA significant and we select the ones containing the plus, we want to keep matching rows this time. So now we can visualize the data using hierarchical clustering. I'm going to stop that activity. Sorry. And I'm going to z-score the data first. So that was this Z button here, and it's also in normalization. Yes, so z-score is subtracting the median and dividing by the standard deviation. So this is, I would say this is a standard normalization. I mean, it's nothing with mean, yeah, not median, sorry. So hierarchical clustering, right. Oops, just a button. So the default parameters, as with a lot of things in Perseus and MaxQuant, the standard workflows are usually good. These two here are in case you wanted to un-z-score the data afterwards. You can report, oh sorry, just this one here, you can report the mean and standard deviation so that you could un-z-score afterwards. I'm not going to do that, so I don't need it. Okay, so hierarchical clustering, same as with the total proteome data.
Obviously, I'm not a statistician, but why are you z-scoring the data?
Okay, so z-scoring basically brings all of the values into the same Euclidean space, and since we're visualizing in a heat map, we only have a certain dynamic range with which we can see large values and small values. So what basically that means is that you bring everything between, for example, -1 and plus 1. So if something's going up by 10 orders of magnitude, it now looks the same as something going up 2 orders of magnitude. That's important for displaying on a on a tool like the heat map, because now you can see the red and the green more distinctly. But it's important to remember that you lose now the relationship between you can no longer compare between the rows of the hierarchical cluster. So you lose the magnitude information, if that kind of makes sense. So, yeah. Yeah, okay, so this is still, yeah. We have 2 minutes or what? I'm not sure what you mean.
There is no strict assumption of normality, but of course z-scoring makes most sense in case you do have a normal distribution across your rows.
Okay, this is probably not finished on your computers, but in the interest of time, I'm just going to show you very quickly some ways to navigate the data in here. So you can, the white space here is not actually white space, but it's where all the gene, or in this case sites, could be displayed. So we can zoom in, and you can see here in this case the UniProt identifiers are shown. By clicking on configure row names, we can change what's displayed there. So we can change it to gene names, and I only want the first gene name so that it's easily readable. And then because it's phospho data, we want to put here position within proteins. So that's the site position. So now we can see all the gene names and the phosphosites. Okay, so if you click on any particular node, you can see here a little red box pops up. So you can click on those, and that highlights the cluster. And you can see then in this box here the profile for that cluster. You can also define clusters here using this define row clusters tool, and set this to different values and see how that partitions your data. And then you can select each of the clusters along the top up here. Now, since it's phospho data, we can also check the since with any data we can check the enrichment here, just like with a Fisher exact test. But it's important for phospho data that we use this relative enrichment tool. So this is specific to phospho because we added protein annotations, but now we're checking if there's a site-specific enrichment. So in the case that we had 1,000 phosphosites on a single protein, we don't want to count that as being significant just because there were 1,000 sites. So by selecting proteins in here, we account for that. So we only count each protein once then. Right? And then you would press this show enrichment box here, which I'm not going to do because it takes minutes. And you would see the enriched categories from all of the categorical columns in this box down here. So, I think that's all we've got time for. But you guys can explore so one more thing I meant to show you is you can, if you hold Control and click on the red, you flip the node. So that can be helpful, especially up here where you've got treatment groups and say I want the control to be first. then you can flip them around like that. Okay. Questions?
Okay. Okay, so I guess then we can have the coffee break.